Yeah, so it's still a theory, the information theory of aging, as I call it. It's emerging in the field, so we're right on the cutting edge, and over the next 5 to 10 years, we'll see where it settles out in the landscape.
So where are we now in the application of that science that you discovered? How do we work through this translocation, basically the movement, if you will, of what were then identified Sirtuin genes, Sirtuin enzymes rather, coded by the genes? How do we get to the place of really getting our arms around the whole notion of Sirtuins, which is, I think when you look in the book under Sirtuins, we see your pictures. So you are the one to talk to about that.
Right. So this sterility of old yeast cells, it turns out that these Sirtuins, these enzymes, normally they control a variety of cell functions, including mating. And during aging, they get distracted primarily by DNA damage. And they leave the place on the chromosome and go and help repair the DNA. But in doing so, and while they're distracted, the yeast cells are sterile. And that was the big finding in the 1990s. When I left MIT and started my own lab at Harvard, I wondered what could be similar about that process going on in our cells, and have spent literally the last 20 years trying to figure that out. And now we know that these Sirtuin enzymes, there are 7 in our bodies, there are 5 in yeast, 7 in ours. They're called SIRT1 to 7. The SIR part of it's interesting. SIR stands for Silent Information Regulator number 2. And the information is important. We'll get back to that, of course. But what we now know is that these Sirtuins can be activated naturally by being a little bit hungry or exercising. Caloric restriction is one way. But we also know you can activate them with chemicals. There are natural and synthetic ones that we've worked on. And also, because these are, at least 3 of them, control the way genes are turned on and off, we call this, gene expression. We think that the changes in gene expression as we get older, in part due to the distraction of Sirtuins, among other things that occur, and it gets worse as we get older and with more DNA damage distracting them. That's, I believe, the major driver of aging, certainly one of the major drivers. And where we're at now are 2 things. In humans, we've got a clinical trial trying to activate the Sirtuins that we have in our bodies already. And then we have what we call epigenetic reprogramming technology that we think is sending them back to where they came from. They're now permanently distracted in a very old mouse and probably a human. And we finally have some handle on how to get them to go back and reset the cell's age and get them to turn on the genes the way cells did when they were young.
So what you're saying is then there's a physical distraction or translocation of these Sirtuin genes aware from, perhaps we could say, they belong to do good things, to going to other places to do other things that are basically repairing damage. For example, we know that double-stranded DNA breakage seems to be a powerful signal for Sirtuin to move away from where it's, shall I say, needed or doing good things, distracting it, and this might be one of the hallmarks of aging, the fact that our Sirtuins are dislocated.
Well, yeah, that's exactly right. And often, including myself, people ask the question, why would the cell be set up this way? It makes no sense. If they didn't do that, wouldn't we live for 1,000 years or more? And the best answer I can come up with that fits with the data is that it's a very ancient system, right? Remember, it's in yeast cells as well. So it's over 1 billion years old. And what I think it's doing is it's coordinating gene control with DNA repair. So when the Sirtuins and other proteins that interact with them go to help repair, the genes that come on are temporarily beneficial. These are stress responses, DNA repair factors. In the case of yeast, becoming sterile is a good thing. You don't want to be mating while you have to repair your broken chromosome. But the problem, I think, is that though the system does reset, it doesn't fully reset 100%. And so one analogy is during Hurricane Katrina, the emergency response troops went down to to New Orleans. And then not all of them went back to their homes, and their homes became full of weeds and didn't pay the bills. And if you keep doing that over, in the case of our lifetime, 80 years or more, then you'll end up with a huge mess in the country. Or in the case of the cell, a lot of genes that are now dysregulated, and the cells lose their ability to function and eventually lose their own identity and forget what type of cells they are.
We're going to get to that. I mean, that's I think that's probably leading edge of where we are. But let me take it back to the notion that the Sirtuin relocates for one reason is to this area of damaged DNA. And there was a time when the accumulation of DNA damage was thought to be the main driver of the aging process. So in a sense, though, your newer understanding of the aging process does kind of envelope the whole notion of damaged DNA and other inputs in terms of Sirtuin relocation.
Yeah, it does. This is what's been rewarding about the data that's come out over time. Of course, there's mutations that occur, and there's lots of DNA damage. I'm from Australia originally, and all you have to do is look at Australian skin, people who have sunbaked, or Floridians, as I mentioned. You can tell that it certainly accelerates the signs of aging, and you can accelerate what we call the DNA methylation clock that way. But unfortunately, antioxidants and DNA repair factors haven't been very successful at extending the lifespan of any species, even yeast cells. So there was some component of this idea that came out mostly in the 1950s and 60s when the world was obsessed with radiation and the effects of it, that's been unsatisfying. And what we've come up with is the proposal that DNA damage is important. And so getting exposed to UV light in the sun, of course, you want to avoid that for numerous reasons, including cancer and aging. But there's this other component that's actually the main result of that process, and that's the changes to the epigenome, the factors that control the gene expression, the cell's identity. And they're intimately tied together. They're both, so the genome gets damaged and the epigenome responds and doesn't fully reset. And over time, they end up causing cells to lose their function. Perhaps, David, it would be good if I explain a little bit about the epigenome.
Absolutely. So, we've heard our viewers are somewhat familiar with it, and maybe we go back to Waddington. Maybe we'll go back to the notion and explore your analog versus digital kind of a metaphor about, and look at the responsiveness, therefore, of our genetic code to our environment. That would be terrific.
Right. Well, actually, so the epigenome was very popular, again, in the 1950s. So Conrad Waddington proposed, in his efforts to explain how is it possible that a fertilized egg turns into an organism, a baby with 26 billion cells that know what they're supposed to do from essentially the same genome. How is that possible? So he came up with the cellular equivalent of a recipe. And what actually happens in physical reality is that the DNA is looped in big loops and spools, and it controls in each different cell type which genes should stay on and should stay off. And there are chemical marks that dictate that this is a gene that should stay on for 90 years in the brain or more to make that cell a neuron, for example, or a skin cell or a stem cell in say, the muscle. But what Waddington had no idea was that this was going on. That they were barely getting a grasp on DNA. They didn't know these other processes. So what he came up was a metaphor, which was that the fertilized cell is like a ball at the top of a mountain that would roll down and not randomly, but be directed into valleys where the ball would rest, or the cell would rest. And that is a stable cell type. And if you're in a valley for a neuron, you cannot easily jump over the hill and become a skin cell. And that leads to the embryo and the baby. but he didn't know what the valleys and the hills were at the time. And now we know it's these proteins and DNA modifications like DNA methylation that create those hills and valleys that last almost the life of the individual. But as I mentioned, in the case of yeast through to humans, we see that those hills and valleys start to flatten out so that cells start to wander over into the wrong valley.
So then what leads to aging then is this sense that the methylations are in not exactly the right place, and that causes gene expression that doesn't bode well for us, leading to things like loss of proteostasis, mitochondrial dysfunction, exhaustion of stem cells, altered intercellular communication, all of the things that people seem to focus on these now as mechanisms of aging. But what you're saying, though, is this is the primary event. This is the upstream event, is this dislocation, this change in the methylation pattern of our epigenome that then it correlates so well with aging as, well, let's talk about the work of Dr. Stephen Horvath then. He looks at various patterns of how these methyl groups look in terms of their disbursement on the entire epigenome, and is able quite handily to correlate that with our rate of aging, our state of aging, pretty close correlation with chronological aging. But it's more or less a DNA methylation aging kind of clock that bears his eponymously is called the Horvath Clock. And so that has, at least until recently, in Horvath's description, was pretty much indelible. I mean, we're kind of jumping the gun here on some of your newer research, but this whole idea that the changes in this methylation pattern really, may I say, represents our aging, and is really the upstream event that then filters down to all of these other mechanisms that researchers are exploring as leverage points to deal with aging.